Thanks for joining me for this video lecture on DNA mutations. In particular, we're going to look at some of the common ways that DNA can be altered and what the results in the final protein product could potentially be. So when we think of a mutation, we're thinking of a change in the DNA sequence that typically, though not always, results from mistakes during DNA replication. At the DNA level, the most common form of a mutation is what we call a point mutation, a mutation that affects only one position in the DNA sequence. There are a couple different types of point mutations. There are, for example, base substitutions, where a single nucleotide is replaced. And so, for example, maybe DNA polymerase is supposed to put an A in a particular position, and it accidentally puts a G, and proofreading doesn't pick it up. And so the G gets carried on to the progeny. We would call that a base substitution form of a point mutation. Other point mutations could include insertions, deletions, and reversions. An insertion is when one or more base pairs inadvertently gets inserted into the sequence. Same with a deletion, one or more base pairs inadvertently getting removed from a DNA sequence. Insertions and deletions, as we talked about earlier on when we talked about DNA replication, will lead to a frame shift. When there's a shift in the reading frame, everything falls apart. We'll talk about those again briefly in just a minute. A reversion, however, typically happens with a single base substitution, though theoretically it can happen with an insertion or a deletion. It's when whatever mutation happened earlier on gets reverted back to the wild type, to the original DNA sequence. And reversions will happen at some particular frequency, and we have good evidence for reversion. So these are the point mutations: base substitutions, insertions, deletions, and on occasion, reversions. There we go. I want to show you some point mutation examples. So in the top one, you can just see a base substitution taking place where you originally had an AT pair and DNA polymerase accidentally puts in a GC pair. And if it doesn't get picked up by proofreading activity, then it gets passed on to the progeny and becomes permanent. Indel mutations, insertion, deletion mutations, indel mutations, are when something either gets added or removed, causing a frame shift. So here's an example of an insertion. So normally we would have GGC moving on to GCT. Now we have GGC moving on to AGC. So you see the GCT codon is gone. And now we've got an AGC codon, and all the codons downstream of the insertion or potentially deletion will lead to a frame shift. They'll all be offset, and therefore everything downstream from an indel is changed. So what impact might these mutations have at the protein level? Well, a mutation could be silent. This isn't at all uncommon. This is when the DNA itself is changed, but the resulting amino acid, because of the degeneracy of the genetic code, is not changed in any way. So for example, if what was normally a GUU region, right, a codon, was changed to a GUG, where the U was mutated to a G, in both cases we get valine. And so the mutation is there at the DNA level, but at the protein level it's considered silent because there's no impact. And so a significant number of these point mutations in particular are going to be silent. A missense mutation is when one amino acid is changed to another amino acid. So for example, possibly valine being changed to an alternative amino acid. If instead of this third position in the codon being changed, maybe it was the first position in the codon being changed. Now, missense mutations can be either neutral, or sometimes called conservative, but a neutral missense mutation is when the amino acid does change, but it changes within the same general group of amino acids. So for example, going from one charged amino acid to another charged amino acid, or changing from a hydrophobic amino acid to a different hydrophobic amino acid. The general properties of the R group are similar, and that's going to minimize the impact on the way the protein folds and functions. That would be a neutral, sometimes called conservative missense mutation. A non-conservative or non-neutral missense mutation then is when the amino acid that changes becomes something completely different. So if we go from a charged amino acid to all of a sudden having a hydrophobic amino acid, that's going to change the property at that location, the primary structure of the protein. It's going to change the way the protein folds and potentially the way the protein behaves as well. Then we've got a nonsense mutation. So silent mutations, missense mutations, and a couple different types of missense mutations, and then nonsense mutations. This is when an amino acid actually gets changed, when a codon gets changed from coding for an amino acid to simply becoming a stop codon. Now all of a sudden you have a truncated protein that's stopped too early, and depending on how far into the protein that is, that could have devastating effects. The protein may not be complete and very likely won't be functional. Now, we've mentioned frame shift mutations a couple times. This is when an indel mutation of one or a few base pairs causes a change in the reading frame. Now, if it's in a multiple of 3, then the insertion deletion frame shift will be very short. Only the amino acid before and the amino acid after, the codon before, the codon after the indel will be impacted, and then we'll be back on track after that. But if it's not a multiple of 3, then everything downstream is going to be off, either creating a very long protein that makes no sense, or potentially introducing a stop codon and making a very short protein that also makes no sense. Frame shifts are arguably the most dramatic and devastating mutations that we see at the protein level. Now, an equivalent reversion, we talked about reversion mutations. This would be where an equivalent reversion would be where the amino acid is changed, but then changed back through later mutation. But that later mutation doesn't change the codon back to its original, just changes to an alternative codon. So, for example, UCC coding for serine gets changed to UGC, which is cysteine, and then later in rounds of division, the UGC turns into an AGC, another way of saying serine. So it would be considered an equivalent reversion. So we went from UCC back all the way to AGC, a very different codon, but the net effect was neutralized essentially. It was equivalent because we ultimately ended back up with serine again. And there are examples of all of these, and you can imagine they happen at a variety of different frequencies. In this final slide, I just want to show you visually what this might look like. We've got on the left different types of DNA mutations, ways that they might affect the protein, and then visually how that might look. So, for example, we can have a silent base substitution where the G here that was giving us a codon that led to proline was changed to a C, and in that case the codon, because of degeneracy, still leads to proline. That's a silent mutation. It might be a missense mutation where this G here, right after our start codon, which GCC codes for alanine, this G gets changed to a C, all of a sudden we've got a proline. So this is a missense where we changed from alanine, all the way to proline. So we changed our amino acid. We can also have a nonsense mutation where AAA led to lysine originally. Now we changed this A to a T. We've got TAA, that's a stop codon, and we've got a truncated polypeptide. And then here's an example of a single insertion mutation where an A is inserted up here, and after our glycine before what should have been proline, we shift everything downstream, and now instead of proline stop or proline lysine glutamine threonine or glutamic acid threonine, we go glycine and threonine, glutamic acid, arginine, aspartic acid, and then on down the line, and we miss either a stop codon or we mess up all of these amino acids, whatever the original was. So you can see the frame shift mutation is far more dramatic than any of the other mutations. The highlights of what we just learned. Changes in DNA are what we call mutations. Common DNA mutations include base substitutions, insertions, and deletions. And these different forms of DNA mutations can have a wide range of impacts on the final protein. Make sure you spend some time learning the terminology. The concepts are relatively straightforward, but the terms can get jumbled up, things like nonsense versus missense, etc. So spend some time committing those to memory so that the language is comfortable and familiar to you. So as we move forward dealing with DNA and mutations, you know exactly what you're talking about. Feel free to watch this video as often as you like. Give me any comments that you need to. Thanks.